Good afternoon, ladies and gentlemen. Mark Caulfield and I have a dispute about whether it's 2:39 or 2:40, so we're splitting the difference. I'm delighted to see you all here for this lecture, and I'm equally pleased to have been asked to chair it. I've known Mark for a long time. He graduated in medicine in 1984 in London, and then went on to train as a clinical pharmacologist at St Bartholomew's Hospital, otherwise known as Barts. He became interested in hypertension originally, in the molecular genetics, and was on a NICE Guideline Group for hypertension, and even became president of the British Hypertension Society, which I occasionally attended in those days, and then became director of the William Harvey Institute at Barts in 2002, although he's now, I think, vice president of that whole outfit, and excellent. Since 2008, he's directed the NIHR. The NIHR is the National Institute for Health Research in this country. It gives a lot of funding, and there's an NIHR, Cardiovascular Biomedical Research Unit and Centre at Barts, which he directs. And he has also been instrumental in some of the reorganization of London hospitals, which has changed the face of clinical practice in London. He has won too many prizes and been awarded too many honours for me to list here. And in any case, I wouldn't embarrass him by listing them all.
Thank you.
But the main thing for which he's known, I think, certainly in this country, is his role since 2013 as Chief Scientist for Genomics England, which has run an enormous outfit detailing what is known as the 100,000 Genome Project, which has delivered incredible amounts of information about genetic disease in this country. And so that's what he's going to tell us about today under the title, Transformation of Genomic Healthcare. Mark.
Thank you very much, Jeff, for that very kind introduction. I'd like to thank the World Congress for inviting me to speak here. On this slide, you can see my hospital on the left, which is 900 years old this year. And on the right, you can see William Harvey, who discovered the circulation in 1628 at the hospital, and that we use that still today to manipulate in the treatment of cardiovascular disease. I forgot to put a disclosure slide in, but nobody's given me any money to say anything, so I have no disclosures. I'm going to talk a bit about how we have used a single project to transform the way in which we do genomics in the National Health Service in our country. This project is a surprising origin. It is an Olympic legacy project from the London 2012 Olympics, where the then Prime Minister, and we've had four since, had the idea of assembling scientific advisors and ministers from around the world, and they told him that it would be a good time for a free at point of care system to test the role of whole genome sequencing in the NHS. And so the project was focused on rare disease, cancer and infection. We enrolled over 97,000 people in the program. But what we did do from the outset, which served us extremely well, was to involve public and participants in a participant panel, which was strategically involved in the decisions that we made and also in supporting us in focusing the efforts in the program. It was about 40 petabytes of data, but today I checked, and it's now 90 petabytes of data, and it is stored in an Amazon cloud that is held here in the United Kingdom. And that acts as a trusted research environment. We kind of invented that concept, which is where you make available a data environment for people to work in, and it acts like a reading library, not a lending library, so you don't give the data away. However, if you come and do research on it, you can take your data away for publication, and the data remains in the embassy, so you can go and inspect others' work and check it's valid. We developed an infrastructure of genomic medicine centres. There were 13, and they covered 98 of the 220 NHS trusts, which are hospitals in England. And this paved the way for point-of-care transformation and the involvement of up to 5,000 frontline NHS staff who worked on this programme across the 8 years that was going on. And this meant that when we ran into trouble with how cancer is preserved in formalin, which is damaging to nucleic acid, we were able to inset 400 molecular fresh tissue pipelines for cancer care across the NHS. And because this was new ground and not many people had done many whole genomes, we opened the data up to researchers who could openly apply, and you can too, to work on the data. And it's available now to researchers from a secure data center in 33 countries. And they have access to a massive data resource, which is growing all the time. I'm going to focus in on rare disease, and I'm coming at this now from the point of view of the diagnosis paving a way to treatment. So if I set the scene, about 6% of the Western societies will have a rare disease. That's about 1 in 17 people. If we go to the Gulf and the Arabian Peninsula and to Pakistan or Bangladesh, this rises to between 8 and 12% because of closely related intermarriage. When we started this program, about half the people go through life without a molecular diagnosis. They may get the name of a syndrome, but they don't know why they're like they are. And what I'll share with you is that one of the things above all else, the parents, particularly of children, or the adults themselves affected by these diseases, crave from you or I, is a diagnosis, because they know without that, none of us can think about a treatment. So I'm going to focus a little bit on a pilot study that we did and published in November 2021 in the New England Journal of Medicine on the first 2,183 families. What we did when we started, I'm not a rare disease or cancer doctor, I'm a heart doctor. I work on blood pressure, and I didn't know much about rare disease when I was appointed and had to learn on the hoof. But one thing that struck me was that the community had not standardized phenotyping and characterization of patients. So we sought to standardize that, but also to maximize the number of clinical terms with which the patients were annotated. And to do that, to speak to other data sets around the world, we use something called the Human Phenotyping Ontology, which is now mapped to SNOMED CT. And that means data can talk internationally, because you may need cases in other countries to solve your small number of cases in your own country and neighborhood. We automated the analytics, and we involved the NHS in the whole process. The picture of the young girl on the right is a child whose mother thought she was going to die. She had, since birth, progressively neurologically declined. She was having real difficulty walking. She thought she was heading for a wheelchair. She couldn't communicate at all. There was no interaction whatsoever. And we found that she had something called GAMT deficiency with a whole genome. And that GAMT deficiency means she can be treated with a simple dietary shift where we increase the amount of creatine, ornithine, and arginine that she receives in her diet. And that child can now talk. She will never recover her full intellectual capability, but what if we could have discovered that earlier and perhaps incepted this very simple, inexpensive change? I will show you a number of other cases where we've applied this to change the therapy. The first is a sad story of a 4-month-old who was born, went straight to the neonatal intensive care, in fact, never left the hospital, and he died with no diagnosis, and immune testing was negative. His parents asked to enroll him in the project because they wanted to understand, if they tried to have another child, what the likelihood of them having a similar outcome was. But then Mum unexpectedly became pregnant and said, look, I don't want to know, don't tell me. But at month 8, she started to get a bit anxious, and so she said, look, actually, I think I would quite like to know. And we found that the deceased boy had a transcobalamin 2 mutation, which are documented in the literature. We're going way back to the 70s. This transcobalamin 2 mutation means the child cannot take vitamin B12 inside cells, and therefore the neurological presentation and the infections that he had that were repeated and very difficult to treat were due to B12 deficiency, something simply correctable. His brother was born now about 7 years ago now, and he was, because they knew where to look, tested in 1 week by the NHS, and he was also sadly affected by the same disorder. But because we knew about it, he received very high-dose vitamin B12 on a weekly injection regime, and that child has had a completely different course. And if he came in here today, you would be unable to notice there was anything wrong with him. The total cost of the first child's care was £80,000. Now, I'm sharing that with you because actually sometimes we forget how expensive it is to look after people who are very, very ill or seriously physically or intellectually disabled. And the cost of a genome, which at that time was about £858, pales into insignificance when you're spending this money. Think about this if you could avoid all of this. So here's a second case. This child was 10 when she was admitted to ITU with life-threatening chickenpox, something many of us here may have had as a child, and it was debilitating, but you were all here. But this child had been multiply admitted to ITU with severe life-threatening infections at a total cost of care of 359,000 sterling. We found mutations in the CTP synthase 1 gene. These affect B and T lymphocyte functionality and actually prevent you responding appropriately to both capsulated bacterial infection and certain viruses. So she received a curative bone marrow transplant at a cost of £70,000, but her siblings were also tested and to the great relief of her family were not at risk of these infections. So again, I show you an example where we spent a lot of money on care when a genome could have unlocked this. And we're just lucky she managed to survive to her 10th birthday to get the diagnosis. This is a child, and I'm going to show you a picture of him. His mum and dad are very keen for you to see him. He's called Owen, and they are very happy for you to know his name. And I've told you that because if you go to the Genomics England website, you can see a video of his story. So this is a child that had been given a diagnosis of diamond black fan anemia, a very serious condition which can often end in cancer. But when his clinical features were looked at, he was anemic, he had developmental delay, he had constipation and short stature. This led us to apply a particular analysis for developmental delay, which detected a de novo mutation in him, so a new mutation in him, that meant that his thyroid hormone receptor alpha wasn't sensitive enough to thyroid hormone. So you can give thyroxine in higher doses, titrated against the metabolic rate of the child, and correct this almost completely. So if you want to look at this, there's a link there to participant stories. You'll be able to see this poor child, when he first was enrolled, was barely able to walk. He couldn't walk unaided. He had to hold both his parents' hands to walk. And by the end of the film, you'll see that he can dance and he can also communicate very effectively, and he couldn't when he started. So that's the difference that these make. Now, I'm not going to pretend to you that this is the case for everyone, and as you will know, many of the people who get a diagnosis, that is the end of the road currently for them. But with the current platform for new therapeutics, especially gene therapies, and as we were hearing this morning, antisense oligos, for the first time there are potential trials available for these people. So everyone in this project is enrolled for longitudinal life course follow-up by electronic health records and recall for research. So every person, if there's an opportunity to engage in a trial, can be recalled and offered that opportunity. And that's still true in the National Health Service today. Just to take you through some facts and figures from that, what we found in the programme is the pie chart on the left is showing you what you can get from a panel in the blue and then what you can add in yellow, green and orange from a whole genome. And you add quite a lot. You get a much more even picture of the genome, of the 3.3 billion letters that make you you. But also what we also learnt in the programme was that the notion that if you sequence more family members, you could get better line of sight on the diagnosis is true. So if you had two family members, usually mum and a child, or two parents and the child, you could get a diagnosis more easily. Diagnostic yield improves with increasing family size. And then when we focused in on those who were likely to have a monogenic cause and added research endeavour on top, we could get across the first 2,183 families to a 38% diagnostic yield. If the disorder was likely to have a more complex etiology, for example, be oligogenic with a few genes contributing, it was much harder to get a diagnosis, and we only made a diagnosis in about 12%. Now, here you're seeing by different systems the disorders, and you'll see that there are a number of disorders, hearing or ear, metabolic, intellectual disability, neurological disorders, and eye disorders, where you are well above 30% diagnostic yield. Remember that all of these people had prior diagnostic unmet need. From testing, normal testing in the NHS, they had no answer for why they were like they were. And if we take this further and move to what happens of a whole genome compared to prior or no prior testing, well, whether you had a test or not, you're about 33% more likely to get a diagnosis with a whole genome than you are with any other type of test. Though I caution that on the right-hand side, you'll see whole exome sequencing indicated as WES, and there's only 16 people there, so you need to be very cautious about that. We might have done better with a bigger sample size. That could be the play of chance. But a whole genome does get you more diagnoses. This is a diagnostic odyssey of children in the program born from 2003 onward. So this was censored at 2019. And so they'd spent by that time a median of 6 years, 75 months, attending 68 hospital appointments. And what you see with these people is that in and out of the health system, sometimes several times a week, and unaffected relatives over the same period had a median of 18 appointments, over a longer period had a median of 18 appointments, so far fewer contacts with the system. Post-diagnosis, what you find is we're able to focus the clinical episodes on the diagnosis and the downstream testing or support that is necessary to help that individual. So they have far less contact with the health system, but it's about what the problem is. And those affected individuals use 183,000 episodes of healthcare in their first few years of life, at a cost of £87 million, compared to 53,000 episodes in the unaffected relatives at a cost of 21 million. So it's not to say that people who are unaffected by these things do not use the health system, but there is much more use if you have a rare disease. And this doesn't include their primary care contacts or their disease treatment costs. So we found that overall, about 25% of the diagnoses had immediate clinical utility in the NHS. They changed medication. They led to additional surveillance for probans or relatives. Some were eligible for clinical trials, and the diagnosis could, in some cases, inform reproductive choices. Or there were other benefits that you could change the diet or give vitamins, and that might treat the condition. Sadly, some of the problems were fixed by the time we made the diagnosis, but you could still improve them in many of the individuals. And we had about 22 new genes that we discovered as a result of this program, but there are up to 829 potential signals that we are still exploring laboriously with functional analysis. This is something that we published. This is Ariana Tucci's, my MRC clinician scientist work. This is on repeat expansions. Repeat expansions are expanded repetitive sequences of DNA. And what you're seeing here is these cause neurologic disease. It was thought that the frequency was 1 in 3,000. So what you're seeing is different neurologic disorders. And what we've applied here is an algorithm, an automated algorithm, to measure the repeat expansion. So this was not thought possible in a short-read genome, but it is possible. And what you can do is just by applying this algorithm, you can diagnose even undiagnosed people who are likely to have a repeat expansion disorder, because these may often take years to present. So I'll focus you in on the bottom row. What you're seeing in the blue histogram on the left is the distribution in the whole of the population. So that's the normal distribution of the repeats. Then there's a red serrated line. That's the boundary between non-pathogenic and pathogenic repeats. The little open triangle is the dad of the boy over near 100, who is shaded in red. That boy presented before his 10th birthday with ataxia and neurologic signs, but because it was an atypical age for presentation, a diagnosis of Huntington's disease was not considered, and he actually then got his diagnosis from this analysis, and then we discovered his dad also had it. But what had happened is there'd been a massive repeat expansion in the child at conception, and he therefore presented much earlier. As we heard from Sarah Tabrizi today, there are now promising treatments for these disorders that may be due to modifiers elsewhere in the genome. Well, our latest work, Ariana, has shown that the prevalence of repeated expansion disorders, which was thought to be 1 in 3,000, is actually 1 in 340 of the population carry one of these repeat expansions. And it may be that modifiers modify the phenotype, and some people just do not express these things, but others do express them, and the expression is dependent on how large the repeat expansion is. So what this message for clinicians is if you see unexplained neurologic disease, think about the possibility of this for a diagnosis. And there are now promising antisense and other therapies in development. So overall, what did we achieve in the whole project? Well, we got to about a 36% diagnostic yield. And since the project ended, we continued to do research on these genomes. So we've made another 2,200 diagnoses since we finished. And that's partly by opening it up to community like you to come and research on it. I'll move now to cancer. Cancer proved to be very difficult. We enrolled 17,339 cancer participants across a range of tumours. The numbers in each group were what we could enroll from the NHS over the time period, and they reflect common cancers. And I'll show you some findings, including work that's in a review at Nature Medicine. So what you're seeing here is a genome arranged in a circle. So you've got all your chromosomes from 1 through to X and Y. And this is a man, and this is the same man on the left circle and the right circle. And this is a gentleman with prostate cancer who was in the program. On the left, his genome is fresh tissue, so it's a biopsy, it's not been put into any preservative, it's been flash frozen and protected, and then we've extracted the DNA and sequenced it. On the right, it looks like a child has scribbled on the center of the genome. And what this is due to the biopsy being placed into formalin, and what that causes is spurious cross-linking between bits of the genome, and so this is completely false cross-linking. So what you also see, if you look at the numbers at the bottom, is that in the formalin-fixed tissue, you have a huge number of variants of all types, and these are mostly false variants. So this is why we re-engineered the NHS to deliver fresh tissue whole genomes. And so here's an example from one of our patients who got into a trial through having her diagnosis made with a whole genome. So she's in the central shaded green circle. The men are squares, and there's a diagonal line through anyone who's sadly deceased. And I focus in on her. Her genome in her tumour contained a BRCA2 mutation, which was also present in her germline genome, because in cancer, you compare the genome you and I inherit from our mum and dad to the tumour genome to find the drivers of mutations that have occurred in the tumour that are driving the cancer to spread, grow. And so when we did her tumour normal comparison, as it's called, we found that she had BRCA2 in both of her genomes, and that allowed her to be eligible for the Olympia trial with olaparib. And subsequently, since completing that trial, she's considered, because this was breast cancer and there was no family history, but she does have BRCA2, she's considered having an oophorectomy for ovarian cancer prevention. More importantly as well was the benefits family-wide, and this family is illustrative of that. Her daughter had a predictive BRCA2 test before her 30th birthday and is now in intensive breast screening with MRI. And her brother and uncles, they have also been screened with BRCA2 for potential prostate cancer risks. So you see the familial-wide potential of the findings to inform others. It may be that people don't want to know, and that right must be respected, but it does give people a chance to be more intensively screened. Here's another case. This is a gentleman who had well-controlled HIV, previous hepatitis B, and what you can see here in his liver CT is a very large colorectal cancer deposit here. And he had biopsy, had usual care testing, which didn't find anything treatable. As a result, he was treated with normal 6 cycles of chemo, but his tumour in his liver enlarged and he was getting discomfort. So we repeated the analysis with the whole genome and found that he has a mismatch repair gene condition, which means he had a hereditary form of colon cancer called Lynch syndrome. And he went, because of this, straight to immunotherapy, and it did shrink his tumour for a while, and he had a better quality of life, but has sadly now perished. So this is some of our latest data. Now, what we're doing here is looking at well-accepted, evidence-based variant genes and the variants in those genes compared to real-world data on survival. So these are Kaplan-Meier survival curves. And I just draw to your attention one here. So this is a cyclin-dependent kinase, which is present in a number of different tumours. The red line are the mutants in the cancer, and you can see just from real-world data, routinely collected from the health system, that they have a far worse outcome. If I go over here to PIK3CA, the mutation in PIK3CA protects you against a worsening prognosis in breast cancer, and we recapitulate that signal. The Kaplan-Meier curve in this signal looks just like the Kaplan-Meier signal in a clinical trial. So real-world data can recapitulate this. And the point I want to make to you is that we should be capturing real-world data routinely on our patients in our trials, because these may allow you and I to detect latent, long-distance, long-term sequelae of benefit or harm. So we can use these trials to follow up our patients and detect longer-term events, and I think we should be doing that. This is to highlight treatment. So there's a problem that can occur in ovarian cancer and breast cancer called homologous recombination deficiency. And here you can see the homologous recombination deficiency, and this is the impact of treatment. If you have HR deficiency, you're more likely to respond to platinum, and in this group here, you're seeing platinum-responsive mutations being effectively treated from real-world data. Same in melanoma, where the people with the high tumor mutational burden have received immunotherapies and are having a much better outcome as a result. They should be the ones with the worst Kaplan-Meier curve. So all of this is from real-world data. Here's another thing that you can do with it. This is a patient with a brain tumor presentation, which was thought initially possibly to be primary brain, but it turned out when we biopsied it that it wasn't clear, so it was a cancer of unknown primary. So we then started to molecularly characterize it with a whole genome, and we found that they had RB1 and NF1 known lung cancer drivers present in the tumor, that the tumor also had a high range of tumor mutational burden, which is more likely to be in colorectal cancer, lung cancer, and melanoma. And then finally, there was a smoking signature in the tumor, so mutations due to benzopyrene exposure, and this clinched it. And this was then possible to treat as lung cancer, where cancer of unknown primary were often uncertain what to do, and the prognosis is bad. So I just wanted to share with you what was done by the NHS with this cancer data. So I'd like you to focus in. This is the West Midlands, which is the centre of England. 3,244 patients. Of those, a Genomic Tumour Advisory Board listed 781 for further consideration. That's 25% of the patients. It turned out that 477 of those had an actionable finding in their genome. What was that? Some had DPYD, which makes you more activate capecitabine and 5-fluorouracil preferentially, and this was at 11.1%. This would led potentially to quite serious adverse reactions. And one of the successes that Munir, who's sitting over there, and I have had is to get DPYD testing preemptively before you receive 5-fluorouracil or capecitabine in this country. And then they got a drug with a clinical license for that tumour. Some were unlicensed. Some majority got into a clinical trial. Some had high tumour mutational burden and could be prioritised for immunotherapy. Some had changes that might be hereditary and therefore referred for follow-up to clinical genetics. So this is not a useless result in the clinic, and it can lead to treatment change, not for everyone. So about 25% of the cancer program got a bit benefit. Now, I'm not going to dwell very much on the next area of pharmacogenomics because it was beautifully covered by Munir the other day, but this is one real success story that the British Pharmacological Society, led by Munir at that time, and the Royal College of Physicians, led by Liam O'Donoghue, who sadly perished during the COVID pandemic, produced a report of how we could implement pharmacogenomics safely in the clinic. So I just thought I'd show you some data from the 100,000 Genomes Project. So here what we've done is to look at 76,000 people's whole genomes. And what we've done is we looked for Clinical Pharmacogenetics International Consortium approved gene-drug pairs that are recommended based usually on consensus evidence for testing in the clinic. And it turns out that 99.5% of the participants had at least one gene-drug pair, where if they come across the medicine, they might either have no efficacy from the medicine or they might suffer harm. And 25% of us have at least 4 of these gene-drug pairs. So this is almost ubiquitous utility of something from a genome for the population. And pre-pandemic, Munir and I were working very actively together on trying to get the NHS to adopt panels so that we could better understand adverse reactions and then move from response mode pharmacogenomic testing to preemptive testing. Here's something that Emma McGavin, who's in the audience, led, it is generally held that there is great underrepresentation of different ethnic groups from around the world and diverse communities in our research. We have a study at Queen Mary at my place, which is looking at British, Bangladeshis and Pakistanis, and has enrolled around 50,000 of them. Emma studied 44,190 and looked at CYP2C19 amongst other genes, and what she's found is that at least 57%, now in white Europeans this is 30%, 57% of the Bangladeshi and Pakistani population will not activate the clopidogrel, a prodrug, and they either really should have some form of point-of-care test or they should have ticagrelor instead. And this work is going to change the way we treat people at our heart attack centre, where we have 2,000 admissions a year and a substantial proportion are from these communities. But this really reinforces what Munir was saying the other day, which is that we do need to study a more broad community of the world to understand the role of pharmacogenomics. And if there's nothing else you take from this, I think pharmacogenomics could have great potential to avoid harm or inefficacy in our patients. And the more we have it, the better things will be. So democratizing research, this is something that we decided to do from the outset, which was to make the data available to any bona fide researcher from any hospital or any institute anywhere in the world. And now, at a peak there were 3,000, but there's now 2,300 working on the data. And they have contributed to 2,200 diagnoses that wouldn't have been made. They published 165 papers and won £55 million in grants. They won that money. That's not Genomics England. And we do work with commercial entities through something called Discovery Forum. But now you literally have access to billions of clinical data points and 140,000 whole genomes, because we've also got the COVID whole genomes, which I'll talk about in a moment in there. So here's the data that you can access in our data center. So we longitudinally life course follow these people. So the data pool goes up by more than a billion data points every 6 to 9 months. And we are following the people going in and out of the health system. This consent for this program is for longitudinal life course follow-up and recall, but it is also to work on any disease or exposure. So in the future, we may be able to do more on pharmacogenomics, but we also may be able to do more on other diseases that people suffer, possibly as a result of exposure to chemotherapy. So how did we use this information in practice? So we use this to transform the NHS, essentially, and create a new genomic medicine service. And with Dame Sue Hill, who is the head of the Genomics Implementation Unit, we established an NHS genomic medicine service, creating 7 laboratory hubs that are regionally based and creating a national genomic test directory. This was quite hard work. So my team and I looked at 300,000 routinely deployed tests in health systems across the world, and we upgraded 25% to new technologies. And one thing that I think the commissioners have only just realized that I did was to build in an annual review of the test directory, which means the tests can only go up. They won't go down as more tests get more evidence and more benefit. And there are 500,000 whole genomes available for NHS clinicians to order against the test directory. And you only get reimbursed in England if you use the test directory recommendations. So it's like a giant NICE guideline, but in the form of a test directory. What this has done is it gives regional equity of access for the first time to genomic testing. And everyone is offered who has a whole genome to take part in research, and they join the UK Genomic Knowledge Base, our National Genomic Library, and 95% of the NHS patients consent to take part in research through this mechanism. So one of the things we have to get better at doing as a community of pharmacologists and clinical pharmacologists is when we're enrolling patients in our hospitals to consider that they might actually really like the idea of doing research and offer them the opportunity to be part of a research cohort so we can recall them and involve them in any research opportunity they might benefit from. There are also some genomic medicine service alliances. This is where the hospital interface is. This is how the test directory is laid out. It's freely available on the web in Excel spreadsheet form. It has a clinical indication, the testing criteria, any overlapping indications, where you use the test in the pathway, requesting specialties that will be routinely permitted to request the test, because in some instances, You don't want the test to be incorrectly requested because it's expensive. And then the technological consideration. Most of the tests are gene panels at the moment, but there are the 500,000 whole genomes available. So what did we do in the pandemic? Well, we turned our attention to COVID and severe critical illness. And a first study that we were privileged to take part in, but didn't lead, was led by Kenny Bailey from Edinburgh. And Kenny enrolled 2,244 seriously ill patients from ITU across England, and he found a number of genes with just genotyping. One of those gene regions was on chromosome 19 near the tyrosine kinase 2, the target for baricitinib. Now, you'll know that baricitinib is in use and in study for rheumatological disorders, but this data, along with the data about baricitinib and how it worked and what its efficacy was contributory to this being adopted in the recovery trial. And in February 2022, the recovery investigators reported in The Lancet that there was a 13% reduction in mortality and length of ITU stay when baricitinib was added to dexamethasone and tocilizumab. So this is building on what colleagues from AstraZeneca and GSK were saying in the Biobank Symposium, that actually you can use the genome to prime this. Now, the genome in this case was not the complete evidence. There was lots of other evidence for baricitinib, and it just helped. But in other cases, I think we can use the genome to prime drug discovery. More recently, in February 2022, we published in Nature this study of 7,491 patients with whole genome sequencing from intensive care, compared to 48,400 controls from the 100,000 Genomes Project. So this shows the utility of having a large genomic database with multiple purpose potential consent. And we found, after you adjusted for genome-wide significance, because you're doing a lot of independent tests, there were 23 signals that were significant. 16 were novel, and 3 were present, particularly in South Indian Asians. That's TRIM46, BCL1A, and LINCO1276. We found multiple gene alterations in expression and nothing in rare variants. There were 2 additional potential therapeutic targets, mucin and GCSF, the colony stimulating factor, which was in trial in COVID. And here are the gene areas, and you could categorize these into inflammatory pathways, immunological, blood groups, and also clotting. There was a variant near factor 8, which may explain the clotting phenotype. We've since gone on to look, as Munir showed you the other day, at vaccine-induced thrombotic thrombocytopenia. Not going to talk about that because he did that the other day. And also, we're looking at long COVID. So we've got about 5,000 people with long COVID that we're looking at. So in 2018, the then Chief Medical Officer, Dame Sally Davis, asked me to do a report on genomic analysis in children because she published her own Chief Medical Officer's report to government, and she wasn't happy with the testing regime for children. So we did 2 things. We assembled a very large, broad group, including public and patients, and consulted on what we should do. And the settings we looked at is genomic testing in intensive care for children, and also newborn screening. So this has led to an exome service, which is run from Exeter, which is now diagnosing at a rate of 44 to 47% unexplained ill children admitted to neonatal intensive care or pediatric intensive care. Now, the importance of this is some of these have had treatable disorders. So we've discovered some biotinidase deficiencies. This is a very simple thing you can correct, but the child's so sick from it, they're in ITU. And the problem is that if children remain in ITU with an unexplained diagnosis, this is very challenging for the parents. The quicker you can get an answer and decide what the course of action is, the better for the child and the better for the parents. So this has been a success, and they're moving that right now to whole genome sequencing. We then looked at newborns, and it turns out that there was no single source of treatable gene changes. So no website you could go to and just get them all. So we made one. This is the work of David Bick, who's at Genomics England now. And it's called RX Genes, unsurprisingly. And it's a one-stop website for treatable genetic disorders. You just plug the name of the disorder you think you have, and it will tell you whether there's a treatment. You see phenylketonuria here treated with a low-protein diet. So you can see what's treatable. So we then analysed, if you did a newborn program and offered a whole genome to a newborn around birth, possibly using cord blood instead of additional venipuncture, of the 4,864 genes associated with the disease, there are 725 disorders that are present in childhood with a treatment. The majority of these treatments are not expensive, only 8% are expensive, and they're transplants or they're expensive drugs like combination chemotherapies or enzyme replacements. We estimate that in 100,000 newborns, 500 to 1,000 will have a treatable disorder. Now, what that means is we will discover the disorder if it's fully penetrant and clearly treatable, and there's evidence that it's treatable. That level of evidence may only be at the level of case reports, because that's as good as it gets sometimes in rare disease. But we estimate that there are 3,750 children born every year in England and in the UK. That's 9 children born every day with a treatable genomic disorder, where if we intervened, we may be able to avoid disability or reduce potential harm. And we calculated that if you do that, you actually gain between 16.8 and 40.3 quality-adjusted life years gain. Now, normally, we're used to looking at data from adults, so you can't usually gain that much life. But if you're a child, of course, you can. So it could make a radical difference, and you can see the funds that might be released from this, because caring for these children, as I've shown you, is extremely expensive. So this has now been funded with 125 million, and the program is starting now. I'll show you in a moment some public and patient consultation that we did on it. So I found throughout this project that involving the people who are affected by the diseases is so important. You might think, oh, that's obvious, but really putting them into everything really changed the dynamic. These were the people who wrote to the NHS when they wouldn't give us the longitudinal life course data and said, hand over our data, and the NHS did. These were the people who turned up at the ethics committee and said, no, we're the ones who put the red pen through this information sheet. We don't want it to read like an insurance agreement. We would not like to sign that. And the ethics committee agreed. So their power and their ability to support us was enormous. And this was a separate activity that we did with members of the public and patients who came together around a piece of work in a series of focus groups. And this is them talking about newborn screening. So they were very supportive of the program, providing it was democratically accessible to the whole population and not just a province of ivory towers. They also thought that if it was confined to treatable genetic disorders, then that would be a good thing. And they were also supportive of the possibility of us retaining people's DNA and information for other diagnoses where there was no treatment, but recalling those people if a treatment or a trial became available. And so the program is just getting going, and it's focused on treatable early life genetic disorders. So we've done other work with the public and patients, and this is work we did with Ipsos Mori around the NHS social contract, and they identified reciprocity, altruism and solidarity as what they thought were the principles that they as patients should bring to the relationship with genomic testing. They had red lines, though. They were worried about genetic manipulation, particularly gene editing, and they were worried about gene editing in their body. They were okay with gene editing of cells taken out of their body and put back in, for example, the bone marrow. They were wary about commercial uses, but they understood that industry was necessary to get the medicines, and therefore what they really meant by this is they didn't want to see their data on television used for marketing by a company in support of their medicine. And they didn't want a surveillance society. And one of the things that I did at the start was to make it an unacceptable use of the data for law enforcement or insurance purposes. And in the UK, we benefit from a moratorium of the use of this data, with the exception of Huntington's disease, in insurability risk calculations. So this is a survey we did on 1,866 people in June 2019, pre-pandemic. And I thought you'd be interested to see, this was used by Munir in the Royal College report, I think, that 72% of the people were interested in how their genetic makeup might affect their response to medicines. That's about the same as those who are interested in current conditions. There's inevitably interest in ancestry and health risks in later life, and some interest in carrier status. So there is a huge interest in the community in this information. But there is also caution about what they might want not to know, which they can't make any difference to. So how have we used this to change national policy? So yesterday, the National Health Service was 75 years old, and we started Genomics England on the 5th of July 2013. So Genomics England is celebrating its 10th birthday. In fact, today it's celebrating it. But I decided I'd rather be here with all you pharmacologists than do that. So this is actually the NHS Genomic Strategy, which was published in late 2022, early 23. And this is the National Health Service CEO writing here in the foreword. And the important thing is that where we've got to is the NHS views embedding genomics through an innovative service model as important with equity of access for patients with cancer, rare disease, common diseases, and we've even, Munir, at last got precision medicine in there for reducing adverse reactions. So we just need another bit of heavy lifting to get this over the line, and PGX will be in there. So this is an unusual situation to be in, where somebody leading a health system has written such a strategy, but that's the impact the programme has had, thankfully, because it was not a foregone conclusion. And so in 2023, the National Health Service is heading towards an equitable and consistent genomic health system for 56 million people. Scotland, Northern Ireland and Wales are also doing things, but they have devolved NHS, so we're working closely with them. And this is standardised, with standardised genomic consent offered to all, delivering against the national test directory and building a single UK knowledge base for research by everyone in the world who wants to do so, who is a bona fide researcher or member of the health system, and with recall for research, so there'll be an opportunity for them. And then it's de-identified for academic and industry use with the ability to recall. But the future is, and this is why I'm delighted to be doing this today, is a global coalition of intellects, and that's, guess what, you, as well as the others who are already working on it, to drive up the value of this for direct healthcare, discover the next generation of therapies, and avoid harm wherever possible in our patients. Jeff, I should like to thank the most important people in that programme. They're not me, they're our participants, and many of them have yet to get an answer for why they're like they are, and they're very much hoping you'll turn up and help them get that answer. I'd like to thank the people who, with me, co-authored the New England Journal paper in 2021. It's a rather large Cecil B. DeMille cast, but that's the way in genomics, I'm afraid. I'd like to thank the various funders that have funded the programme. I'd like to thank the team that did the COVID-19 work that we published in Nature, and all the funders, the Department of Health and Social Care, MRC, LifeArc, Illumina, and the National Institute for Health Research, who funded most of the programme. And I'd like to thank you. If you want to know more, it's here. Thank you.
Excellent. Many thanks, Mark, for a wonderful tour d'horizon of a subject that at last seems to be offering us clinical applications for genetics, which we've been waiting for a long time. It's great to see. There are microphones there and there. Please ask questions. Before while people are getting their minds sorted out, let me ask, is there likely to be any application of antenatal genomic sequencing in the unborn child?
So there is a non-invasive prenatal testing program, which is based on exome sequencing and run from Great Ormond Street. That's separate. Genomics England didn't enter that space because that was already advancing separately on its own. So we really chose to focus on the things where there was a lot of unmet need, Jeff, not because that was already proceeding down a particular line. So there are no plans to do whole genome sequencing prenatally at the moment, but there is a service that is doing prenatal testing coordinated from Great Ormond Street by Dame Lynn Chitty.
Right. Yes, over here.
Thank you. Hi.
Hello.
Kelly Charles from University of Sydney, but a former Barts QUL academic as well.
Nice to meet you.
I'm interested with the rollout of Genomics England into, I guess, a wider population now. How are we, as pharmacology educators, going to start to increase the number of people who are needed within the clinical service to help provide information about genomics, and what does this mean? Because it can't just be reliant on the clinician. There's going to have to be the support network of people that support families as they find this information and what to do next.
I completely agree with you. And Munir, who's at the other microphone, he's done a lot of work on trying to make the argument for expansion of clinical pharmacology, particularly centred around the ability to coordinate and standardise decision support for PGX. Because, you know, like in Australia, I don't know about in Sydney, but in our GPs of about 7 minutes to see a patient, really. And they've got to, so if we drop this from a great height, Kelly, it will never be implemented. So Munir's working very actively on how we could do decision support to support that. I think we do have to embrace pharmacogenomics in our curriculum, and also what we need to do is to make sure that pharmacologists, and I mean I think all clinicians, but pharmacologists particularly, be it basic or clinical, are looking at getting their head around the application of advanced artificial intelligence type approaches to unlock data buried in large data assets, which will, I think, become available from routine healthcare, because that could unlock the next generation of the next industrial revolution in drug discovery. I don't want to overhype it, but I think there is potential there, and I think the genome is going to contribute more to our endeavour in the next few years than it has heretofore.
Thank you.
Thanks, Kelly.
Yes, I was commenting in one of the symposia this morning when I reviewed AI and pharmacovigilance. The work is largely being done by computer experts, very few clinical pharmacologists. We need to get into this field, tool ourselves up to understand AI and start collaborating with all the computer scientists if we want to make an impact.
I couldn't agree with you more.
Munir.
Thanks, Mark. Great talk, as usual. Can I ask two questions?
Yes.
The first one is that the people who've had the whole genome sequencing, there's an awful lot more data available that's not been passed on to them, which may be clinically actionable. So I just want to understand what the contract was with the patients when they had the whole genome sequence done, and what is the legal liability. For example, if somebody suddenly gets carbamazepine and has a risk HLA allele and dies from that, where does the legal liability lie? That's number one.
Yes.
The second one was about industry and working with industry, and I note that your survey, which was very helpful, people didn't really want industry to access some of that data and so on, but how have you tackled that? Because I think it's really important that we do work with industry, because we, academics don't develop drugs, but industry do, and it is really important industry has access to the data.
So to do the last one first, I may have slightly by accident misled you. They were fine with industry working on the data. They didn't want the data used to market drugs. They were fine. They understood that the data was necessary to generate medicines, but they didn't want to see it on the telly as an advert. It was as simple as that. They were not against industry. They recognised that we can't make drugs without industry. So they spontaneously came to that conclusion themselves. On the first one, we agreed a set of parameters which were about actionable findings related to the presenting condition that would be returned through the National Health Service. So the actual point of indemnification is in the National Health Service, but at the point of clinical care and not in Genomics England. So we are not liable for that, but it is, if we make an error, then we could be possibly liable for it. That's why we're extra careful, and all of the findings that we send back are validated usually by an orthogonal test before return to patients. In time, because, you'll know this, but if you have enough whole genomes, you can see which variants you can reliably call 99.999% of the time, and they're never dodgy quality assurance. So those, I think we shouldn't validate them. I think we should just give them back. But for ones where there could be doubt, then validation is still in use. So that's an extra balance and check. So our return of data is confined to those things. We have discussed with the participants. We did additional findings, a limited number of additional findings. So about 1,000 of the 90-something thousand participants had an additional finding, either a BRCA 1 or 2, or a mutation of the LDL receptor or something that we call familial hypercholesterolemia or possible risk of Lynch syndrome. And they have received that as additional findings feedback. But that was strictly limited by the NHS as to what they could do. And I haven't won the battle on PGX, as you'll have guessed. So we just have to keep going on that one, I'm afraid, my friend. Is that okay?
Yeah, thank you.
Great, thanks.
Okay, well, I think there's actually just enough time for us all to get... Oh, there's one more? Have you got... Yes, please.
May I?
Okay,
Thank
last
you.
question. We'll have time to walk to the oral communications.
Quick question. Thank you. Viviana Giannuzzi from Benzi Foundation, Pharmacological Research Foundation, Italy. My question is more or less related with the previous one. Were parents allowed to granular their consent, for example, consenting or not, for the commercial uses of their data, and to, for example, for minors to come back once they reach the legal
Yes.
age to come back?
This is a very important question. So the information sheets, they're all on the web. You can look at them. They they indicate very clearly that industry would have access to the data. Mum and dad gave consent for all children. As soon as the children reach a certain level of maturity or their 16th birthday, they are reconsented. And if they don't reconsent within a 2 to 3 year window, we suspend their data from use in research. So what we've found is that young people between 16 and 18, their minds are elsewhere, they're not so interested in it. But when they get to about 18 to 20, they start ringing us up and saying, you know that thing I did, can I still do it? So they come back. But we had extensive consultation with teenagers from between 12 to 20 on how to do this and get it right in modern age. Because obviously somebody looking like me turning up and saying it's a really good idea to do this is, well, it's not unlikely to go very well. So what we did was to get young people involved, and they shaped the entire approach. So we do reconsent everyone. It's not lawful for them to continue in research when they have attained the ability to decide for themselves, and you have to withdraw them if they don't.
Thank you very much.
Thank you.
Good. Well, thanks very much to the audience for coming and for questioning, and thanks exceptionally to Mark for a superb resume and review of a very exciting area. Thank you again.